Proteins are very important component of a cell. And in the molecular biology, attention is given more to the proteins because the nucleic acid or the DNA or the genome eventually produces the protein. And if we can analyze the proteins, we can analyze and understand the function of the cell or the cellular components more. So in this module, we will pay our attention to the methods which are available to analyze the protein, how proteins are analyzed. Now, when proteins are separated and quantified, they have to be identified first. So what it is, for the proteins, although you separate and quantify them, that is, you determine their proportion, but before you do that, you must identify them. That those proteins, they should be identified. We know that which proteins are they. Now, the best method in order to identify the proteins is that the individual spots on the 2D gel electrophoresis, they are cut out of the gel and cleaved into the peptide. You see, because those spots, they indicate the peptide or the protein. And we have to separate those proteins into the building blocks of those proteins which are peptide. And how we are going to do that? We break the protein molecule into the peptide with the proteolytic enzymes which are available, and then we study them. The technique which is used in order to identify those protein molecules, that what is their weight, how much they are composed of, one is known as Markov mass spectrometry. This is a very popular technique, and it is popular technique known, and the idea or the Markov, it stands for the tidal matrix. It stands for the matrix associated laser desorption ionization time of flight. That is the Markov mass spectrometry. That is, during the spectrometer, we determine the weight of different protein molecule. In this case, what is done? That is the peptide which we obtained previously. We put that peptide on a matrix, which causes the peptide to form crystals. So we make the crystals of the peptide, and then that the peptide on the matrix, it is ionized with a laser beam. A laser beam is put on that matrix and it is ionized. An increase in voltage at the matrix is used to shoot the ions toward a detector. There is a detector, and those ions, they shoot on that detector. And for example, in this picture, you can see that there is a sample. It is put on a matrix. The laser pulse that is bombarded, then got the ion beam which passes through the electrostatic ion reflector, goes to the detector 1, where the signal goes to a computer. And again, from the electrostatic ion reflector, the fractionated or the focused ion beam, it again is put on the detector number 2, which also gives the signal. And by determining all these ions, they have just one charge. That is the one important thing, that all the ions have one charge and then the time it takes an ion to reach the detector depends on the mass. So the principle is that when the ion it reaches to the detector, it depends on its mass. Heavier it is, the more time it will take. The lighter it will go in the shorter time. Just as the higher the mass, the longer the time of flight of the ion. That is, it is the flight of the ion which goes over to the detector. Now, in a MALDI-TOF mass spectrometer, the ions can also be deflected with an electrostatic reflector that also focuses on the ion beam, as I have indicated earlier, that there is another deflection from electrostatic reflector which also deflect the ion beam to a detector number 2. Now, another method which is used is a collision-induced dissociation, abbreviated as CID. It is not CID, which is a police or all that, but that is the collision-induced dissociation. So what happened in this? That ion can be broken at their peptide bond by a process known as collision-induced dissociation. That there the ions are broken by that with a peptide bond. They add their peptide bond by a collision ions, and that breaks the peptide into the smaller unit, into the ions, and then those ions, they go to the detector and give us the mass. Another technique, that is the MS, which stand for the 2 mass spectrometry steps in a row. That is on the peptide 2 mass spectroscopy steps they are performed, and that is known as MS technique. So you see that in order to determine this, there is a MALDI-TOF, there is CID, and that is MS technique. Now, by comparing the masses of ions, which are differing by just one amino acid, if there is one amino acid difference, the nature of the last amino acids can be determined by this method. That is, by the MS technique, we can determine the amount or the one amino acid which is not present in this. As you can see in this, that this is a sequencing diagram of a peptide by mass spectrometry, MS, where there are different amino acids which are present, and it gives the different fraction. Now, the thing is that knowing the sequence of protein, it is very important. We can determine the sequence, the amino acid, where they are present, how they are arrayed in that. But it does not necessarily tell us that the protein, how active it is. So it means the sequence determination is important. Now, but it does not tell us the activity of the protein, that how the protein is active.